Welcome back to Vision. So in the second lecture we have a couple of videos I think will be helpful to give you an idea of some of the visual phenomenon that we see. We'll be talking a little bit more about the nuts and bolts of vision and just also look at how visual information is processed once it actually gets to the brain. So I mentioned the fovea a little bit in the last video lecture. So the fovea is a central portion of the retina, and it's packed with cones. And because of this, it's the center of our gaze, I should say. So whenever you're looking at something, you'll notice that your eye moves so that there's the center part of your eye always centered on whatever you're focusing on. That is your fovea. So why is this important? Well, it's important because since cones have the highest acuity, the fovea is important for being able to detect and see fine details. So, for instance, I actually have a nice dual computer screen set up here. So I have a screen right in front of me here and one over on my side. Now, when I'm looking at the screen on my left, I can't pay much attention to the one on my right. Well, I can pay attention to it, but I can't see the details. If I'm reading to the one right in front of me, I can't read anything on this right side. And that's because my fovea is focused here on this screen in front of me and not on the screen in my periphery. And since these are primarily cones, I don't have that the fine detail perception that I need in order to detect information when I'm not focused on it with the fovea. So fovea is very important for detecting fine details. There's also the optic disc. So the optic disc is the place where the blood vessels come into the eye, and therefore it's actually devoid of all photoreceptor cells. Because of this, it's a blind spot in our vision. We don't typically see it because the brain is really good at covering up little blips like this, but it's there. So I have a video I want to show you about blind sight that I think will be helpful for illustrating this.
So you've probably heard that you have two blind spots, one in each eye. And this is true. Say this is what your left eye sees. It's a little bit angled because of your nose getting in the way. This is the center of your vision. There's a spot right about here where you don't get any information from the outside world. Your eye is just blind there. Now luckily, your right eye's visual field does overlap at that point, and vice versa with your left eye. So if both of your eyes are open, usually your brain can just cross-reference and figure out what's missing. But if you close one of your eyes, your brain is essentially forced to make up information for this spot here, where nothing's coming in. And it's pretty good at that, but if there's something entirely contained in that area, and there's no clue that there's anything going through it, your brain won't see anything at all. And if you haven't done this yet, you can try like this. You're going to close your right eye, and you're going to stare at this cross with your left eye. This will be your target. Now, if you get yourself the right distance from the screen, just go towards and away from the screen slowly while focusing on this cross, suddenly this target here will disappear at a certain point. And that's the point where it perfectly coincides with your blind spot. So go ahead and try that. Here. And you can do the same effect by just closing your left eye and looking at your right eye over here, and this will now disappear at the right distance. So why do we have blind spots? The answer lies in the structure of your eye. Inside of your eye there's a layer of photosensitive cells that you can essentially think of as the film. They respond to incoming light and give out signals which allow you to see. They communicate with several layers of neurons that begin to process this information, and the information is then sent out to the brain through the optic nerve. Now also back there is a bunch of vasculature, a bunch of little capillaries and blood vessels that are feeding these cells to keep them alive. So if you were designing this system, you'd probably expect light to come in from this direction, since that would make the film out front, this stuff wouldn't be in the way, and this could go to the brain. But in the vertebrate eye, this is actually the back, and incoming light actually comes from this direction. Due to this, there's two problems that arise. First of all, the information at this end, which has been processed by the layers of neurons, has to get to the brain. But since this is the back of the eye, the brain is actually back here. So in order to get to the brain, this information has to go through, essentially, the film. And what happens is there's just a spot on the film where all of the information goes through. And that's called the optic disc, and this is called the optic nerve. And also, that is where all of the blood vessels come through. So basically there's just a hole where all the wiring comes through so it can get to the other side. And that is why you have a blind spot. Now the other problem that I mentioned is that although these layers of neurons here are essentially transparent, the blood inside the blood vessels here is not. And it actually casts a bunch of little shadows that interrupt the image. It's these shadows on the retina and the fact that we can't see them that's the subject of this video. But first I'm just going to mention that we don't actually know why the retina seems backwards the way it is. There might be some advantage to having it this way, but we know for a fact that it's not impossible for it to work the way we would expect it to work, which is with the photosensitive ones out front and everything else in the back. In fact, cephalopods, that's squids and octopi, have that arrangement in their eyes, and so they don't have a blind spot. So going back to this network of crazy shadows that you should theoretically see everywhere you look, why don't you see them anywhere? The reason is sensory adaptation. And this is kind of the same process that causes you to forget you're wearing a wristwatch, or to stop hearing the refrigerator humming after a while. Whenever the brain receives the same information for a long period of time, it basically stops processing it. So since this network of shadows is pretty much always on the same exact spot, since light is always coming from the front of your eye, your brain just doesn't see it anymore. You literally don't have signals about those shadows going to your brain, and therefore they're invisible to you. But that being said, you would then expect that if you look at anything that's not moving for long enough, you would just stop seeing altogether. So why doesn't that happen? The reason for that is that your eye is never truly still. So even when you're staring at something, like this pen, for a really long time, for as hard as you can, your eye is still making tiny, tiny little jerking motions in different directions, and that kind of refreshes the image on the back of your eye. It moves it around a little bit so the different parts see different things. And your brain sees this as changing information, so you don't lose all of it. But if you ever stared at something for a really long time, you might have noticed that you tend to kind of get tunnel vision. You start fuzzing out what you see on the periphery of your vision. The reason that happens is due to the way your eye samples space. You can think of this drawing as the back of your eye seen through your pupil. So the drawing I drew earlier would be from the side, and this is from the front. So this kind of represents the density of how your eye samples space. So towards the center where you focus, your eye has a very dense sampling, so it's very sensitive to changes here. And towards the outside, it gets more and more sparse. So you have larger and larger areas of visual space that are represented by less information, essentially. You can kind of think of them as pixels, and so the resolution of your eye essentially lowers as you get away from the center of your eye. And you'll notice this if you ever try to read something, even if you're an inch or two off of the word you're trying to read, it becomes completely impossible to read it because your resolution is just too low. You can't make out what it says anymore. And because of this also, the tiny little motions that your eye makes aren't quite enough to refresh these guys out here. So although the contents of each of these guys changes with each little jump, so they're refreshed, out here, the really large areas that are all sampled by one pixel, essentially, they don't really change that much. So they might still be seeing the same thing, and eventually your brain starts to kind of tune them out. And that's why you get this tunnel vision effect, where you can still see pretty well in the center, but you kind of get this graying effect. So let me draw in the shadows that you expect to see. You have your optic disc here, and coming from here is a bunch of vasculature, little capillaries going around, and they tend to avoid the center of your eye so that they don't ruin your spot where your vision is the best. So they kind of go around like this, and you have these little tiny fractured lines. So theoretically, if these shadows were to move, you would be able to see them because they would break out of habituation and your brain would think, that's some new information, and you would suddenly be able to perceive all these crazy lines all over your vision. And the way that you would have to do that is to get the direction of the light coming into your eye to change rapidly so that it moves, the shadows shift a little bit back and forth, and you're essentially replicating these little tiny movements your eye makes all the time in order to keep seeing anything at all. And this is actually possible, and it's the reason I made this video. It's a really impressive effect, and I'd love for you all to try it. You're going to need an index card or your finger if you do it just right. What you're going to do is poke a tiny hole in an index card, just a pin or the head of a pen, anything, or use your finger and make a tiny hole like this, right? And you're going to hold it up in front of one of your eyes, close the other eye, and look at a bright surface. You can look at a computer monitor or something that's just blank. Some white paper should do just fine. And you're going to hold it up to your eye, remember to close your other eye, and look through it and shake it back and forth about that fast. You're going to want to shake it so that the hole moves less than the diameter of the hole itself. So you don't want to be going like that. You want to just move it so that you have some spot that you can always see, but the hole itself is generally moving. So what you're essentially doing is making a source of point light that's moving back and forth in front of your eye, and it should change where the shadows land on the back of your retina. And if you do it just right, and you're looking at something nice and bright and blank, you should see this little network of lines. There'll be kind of a light gray, like spider-webby texture across everything. And remember that since you're moving it this way, the light is going to move like that, and you're only going to see things that are aligned in this direction. Because if you have a shadow this way, and you're moving the light back and forth this way, the shadow will move up and down, but it'll still make the same shadow locally, so your eye won't notice a difference. So what you can do is move in little circles to get the best visibility, and you'll theoretically be able to see things at all different angles. And that's how, thanks to the fact that your retina is inverted, just using a light source, a tiny pinhole, and a little bit of science, you can see the vasculature at the back of your own eye. And I think that's pretty freaking awesome. I hope you enjoyed it. Let me know if it doesn't work for you, and I can try to help you sort out what you're doing wrong.
So hopefully that video was fun for you to see. It is interesting getting to see how your perception works and how information gets to your brain and is processed. So I thought that was kind of interesting for you to see. So now we're going to talk about how the information gets from your rods and cones into your brain. So with this, there are a couple of bipolar cells that are worth knowing. On-center and off-center bipolar cells. And these guys work opposite each other, kind of, they're inverse to each other. So on-center bipolar cells actually become active when light is on them. So they receive less glutamate, which excites them and causes them to fire. So again, this is an opposite response. A lot of things are opposite when it comes to vision. So in vision, less glutamate actually excites. So on-center bipolar cells, when the light is on them, causes them to get less glutamate, which causes them to excite and fire quickly. Off-center bipolar cells are actually active when the light is off, so they respond normally to glutamate, and with this they become depolarized the more they get it. So that's how these work in opposite, is because they respond to glutamate differently. Again, for the on-center bipolar cell, when there's light, these become active because there's going to be less glutamate. Remember that when there's light, when the photoreceptors are detecting light, it actually slows down the amount of firing. Or, well, these don't fire, but it slows down the rate that neurotransmitters get dumped into the synapse. So that's what excites these on-center. So that's why they're backwards. It's because they need to react in an inverse way to this. So less glutamate makes these excited and fire. Whereas for the off-center bipolar cells, these become activated when the light goes off because they respond normally to glutamate. So when there's more glutamate, they end up firing more. When there's less glutamate, they fire less. And then these both travel to the ganglion cells, as we'll talk about coming up here in a moment. So this is a very important process. This is how the brain, how it kind of shifts from being backwards to being the way that all other brain information is transferred, as far as with more glutamate being excitatory. It's once the on-center, once you get past the on-center, things start getting back to normal. So again, kind of a crazy system here, so review it. The main thing to know is that the receptor cell will be sending more neurotransmitter when it's not detecting anything. So with that, since it sends less when it's detecting light, then the on-center, which only really activates when there's light, actually has to fire when there's less glutamate. So that's where it's a little backwards. So the bipolar cells that we just talked about synapse with the ganglion cells, which then form the optic nerve. Each optic nerve contains the visual information from one eye. But with this, it actually contains part of two visual fields. Because of this, half of the visual field from each eye shifts at the optic chiasm, resulting in the information from the left visual field going to the right side, and information from the right side, or right visual field, going to the left side of the brain. So the optic tract then terminates at the lateral geniculate nucleus, which is part of the thalamus. So again, we see the thalamus involved in sensory once again. And then this information is transferred to the visual cortex. So the primary visual cortex, also known as V1, is the first place that the visual information arrives after the LGN. The information is then sent to other areas within the visual cortex. The visual cortex is mapped so that certain areas appear to be devoted to certain parts of the visual field. Because of this, damage to these areas can lead to a lack of perception in that area. This is called scotoma, and it's a spot where nothing can be perceived. It isn't a black spot, but rather a blind spot where the brain fills in the information. But that information could be incorrect. So a blind spot, for example, that we talked about with that video, is an example of one of these scotomas. So I want to spend a little extra time talking about the different visual cortexes, because while the book is very good overall, I don't think they did a great job at being clear with this chapter. So as we mentioned, the visual information comes from the LGN to the primary visual cortex. So it first goes to V1, which is primary visual cortex, and V1 is highly specialized for processing information about static and moving objects, and it's also excellent at pattern recognition. V1 is also responsible for detecting fine details and focusing on information from the best eye. After V1, there are actually two pathways through which information can go. The dorsal pathway, which you see in green here, and the ventral pathway, which is in purple. The dorsal pathway will go through V2 and then up into V5, and it's sometimes known as the wear pathway, as it's associated with motion, representation of object locations, and hand-eye coordination. The ventral pathway goes through V2 and then to V4, and it's sometimes called the what pathway, as it's responsible for recognizing objects and storing information into long-term memory. The dorsal pathway is also responsible for the phenomenon that is called blind sight, and I have a video for this in just a moment to show you a little bit about blind sight. So, returning to the visual areas, V2 is responsible for orientation and spatial frequency. V2 cells can also respond to illusory boundaries and perceive complex relations among parts of receptive fields. So that's where this picture comes in. So if you look at this, you probably can see a triangle here, and you probably can even see a triangle here. Now, the boundaries aren't there. There aren't lines connecting these. So the fact that you see these two triangles is actually part of V2, where it's able to see or detect those boundaries that don't actually exist, but putting the information together so you're able to see those boundaries even though they're missing. So V2 is a place where information is combined, so we can look at differences between the eyes and gain a better understanding of where things are in space. So this is an important distinction to make. In V1, V1 on each side just has information from each eye separately. V2 is the first place it's combined. So that's part of how it works, is this information is finally coming together from both visual fields and then can be utilized together in tandem. So with this, V3 is thought to be part of the dorsal stream, and it receives its information from V2. It plays a role in motion perception, though not nearly as much as V5, which really does a lot with motion, as we'll talk about. V4 is part of the ventral stream, and it is responsive to orientation, spatial frequency, but it's best known for color perception. And then lastly, V5 plays a major role in the perception of motion. So now we have a blind sight video that I think will be helpful and kind of gain a broader understanding of these two pathways.
Graham is one of the world's few known blind sight patients. He is blind, yet he can see. This paradoxical condition shows just how much our brains get on with running our lives without us even being aware of it.
When I was 8, when I had the accident, it was a road accident that caused the brain damage, I literally used to walk into lampposts. I ran into, you know, these huge great pillars you get in stations. I ran into one of those one day.
The road accident caused colossal damage to the main visual centres of Graham's brain. He can see to his left, but he is blind to everything on the right, in both eyes.
If you put an object in that part of the field and ask him what is it, he has no idea. He cannot perceive it consciously.
Down.
And yet the remarkable thing is, if you move this object, he will tell you which direction it's moving.
Right.
Even though he cannot see the object.
Up.
Blind sight is this term introduced by Larry Weisskranz to describe the ability of people like Graham to detect things but not to be aware of them. So very, very different from what we would normally call vision.
Colin Blakemore is one of several Oxford scientists for whom Graham's mysterious abilities raise baffling questions about consciousness.
I mean, blind sight is extraordinary when you see it. It's shocking. I think it's shocking because it brings home the fact that we can actually manage our brains without consciousness to some extent. And that leads to the question, well then,
Why not everything?
why not everything? And why do we need consciousness for certain things? What is the extra gloss that consciousness gives, if anything, to our actions?
For nearly 20 years, Graham has been the willing subject of a range of experiments that have yielded astonishing results.
You can see things over here.
Oh yes, I can see.
I'm going to move my hand across. You tell me when it appears, when it comes into view.
Now.
Very precisely as it enters the seeing part of your field.
Yeah.
And if I just hold it over here and you look there, you can't see anything?
No.
How about now?
You're moving it up and down.
But you're seeing it.
It's very easy for me to say to you, oh, I saw that move up, Colin. And as soon as I say that, you're going to say, ah, he can see. No, I can't.
Okay, let's see if we can just test that a bit more formally. What I'm going to do is make squares that appear on the screen, and they will move either upwards, downwards, or out to the right. You've got to keep looking at this point all the time, so it's in your blind visual field that this is happening, where you're not supposed to be able to see anything at all. But what I want you to do is just to guess what the direction of motion might have been, even though you can't see it. I want you to tell me each time whether you're aware of anything having happened, okay?
Down, not aware. Down, not aware.
Not only does Graham see nothing on the blue side of the screen, he's not even aware of anything happening there at all.
Not sure of that one. I think it moved to the right.
Yet miraculously, he guesses the direction of movement correctly.
If there's one thing that this phenomenon of blind sight teaches us, it is that vision is not entirely seeing, that there can be a disconnection from the capacity to respond to visual information and the actual act of being visually aware of something. Those two things can be separated and probably are in our everyday lives. But the problem is that obviously we're not aware of the things that we're not aware of. We just don't know the extent to which they play a part.
So pretty cool stuff. So again, that's showing you the difference with those pathways, where in Graham's case, the patient's case, the ventral pathway was damaged, and was damaged probably at V1, because with that you're losing information from one whole side of the visual field. So that would explain it's probably V1 on the left side, which is causing damage to the right visual field. But with that, it shows that there is that other pathway that information can travel, and that and be processed, even if it is outside of conscious awareness. So really pretty cool stuff. So Also, there are three basic dimensions of color that we should talk about. These are brightness, hue, and saturation. So brightness varies from light to dark, and you can kind of see along this spectrum here, so from light to dark. So that's brightness. You have hue. This varies throughout all colors. So what you see is the different colors, those are different hues. And then saturation differs from full colors to gray. So you can see higher saturation to lower saturation here. So there are a couple theories of color vision that we'll just touch on briefly. First, there's the trichromatic hypothesis, and this is that there are three different types of cones, and that each respond differently to different parts of the spectrum. And then each one has a different pathway to the brain. So it's explaining the fact that we can see three different colors based on that we have basically different cones that cover these different parts of the spectrum. So it's kind of a range fractionation argument. There's also the opponent process hypothesis, which is that color vision depends on systems that produce opposite responses to light on different wavelengths. So this accounts for the fact that, as you will see in a second, that cones actually see a range of colors and thus identify one color that's being seen, whereas another is oppressed. I think this is most easily illustrated with a example. So one thing that always kind of rings true to me is, have you ever had it where you're driving and you accidentally looked at the sun for a little bit, and then there's this like dark area after that, after you look away from the sun, there's a dark spot in your visual field for a little while? That cannot be explained by trichromatic process or hypothesis. That would be explained by opponent process, where essentially what happens is there are these opponent colors, and while one is being exhibited, the other is being oppressed. And if that flips, as you have happen when you look away from a bright color, then you see the other color because it's no longer being oppressed. So both of these hypotheses have some support, and neither is fully explanatory. It's true that it looks like the different cones are responsible for seeing different colors, and that's part of the reason why some individuals are colorblind. On the other hand, some cells in the lateral geniculate nucleus do show this opponency, meaning that for different colors they have opposite effects on how they fire. So there's some evidence for both of these hypotheses, but neither is fully explanatory of why we see color. So here we just see kind of a description of them. So what you see first is trichromatic over here, where you have a couple different types of cones, and you see that they're responsive to different parts of the wavelengths, or different parts of the spectrum, rather. So that's how you see the different colors, is because different ones are responsible for seeing different aspects of it. And then over here with the opponent process, what you see is when one color is being excited or being expressed, it causes the others, the opposite, to be suppressed. So that's opponent process. And again, there's evidence for both, and neither fully explains our color perception. Now, as we talked about a little bit before, there are two main processing streams for vision that originate in the visual cortex. There's a ventral processing stream, and this is important for identifying objects. So this is the what pathway. So here's your ventral stream. So it's important for recognizing faces, objects, things of that nature. You also have this dorsal stream, which is important for detecting the locations of objects. So this is your where stream. So damage to either processing stream can cause impairment. For instance, optic ataxia can cause difficulty in using vision for reaching for an object. And this would be, of course, damage to the dorsal stream, which tells you where objects are in space. And then damage to the ventral stream can cause either difficulties in identifying objects, or this is more of your conscious vision, so it may cause vision loss as well. Myopia, or nearsightedness, is what a lot of us have. That's where you have difficulty seeing things that are far away. So myopia develops through continual focusing on close objects that require the lens to be unrelaxed. It appears, well, with this, it appears to be due, at least in part, to focusing on close objects too much during childhood, though certainly there is some genetic component to this as well. And it results in a lengthening of the eyeball, which causes the image to converge too early, thus leaving a blurred image on the retina. And then corrective lenses, of course, can be used to bend the light so it comes together at the right point, right on that retina. Also, a couple things that can be preventative of this. Having adequate light, especially for reading, larger type, and then also reading without glasses can all help prevent myopia. Unless you're too blind to read without glasses like I am. There's also amblyopia, which is a reduced visual acuity that's not caused by optical or retinal impairments, and this may be due to the eyes not being properly aligned or other problems that causes one eye to be weaker. The visual cortex covers this up by using information from the dominant eye. In childhood, if this persists over time, the eye can actually become functionally blind because with brain plasticity, it will reallocate those visual areas. However, this can be prevented through either early surgical intervention that will make both eyes be good as far as information, or blocking the good eye for periods of time, forcing the brain to still use that weaker eye.